Hey everyone, are you new in the field of bioinformatics and struggling with molecular docking, but your ligands are stuck as flat 2D SDF files? Then this video is for you. Here we see that you cannot run accurate docking without proper 3D coordinates. So don't skip the video. At first open the browser and search in the search bar, that is PubChem databases. From the PubChem databases, the ligands are found. Here we opened the stearic acid. If we click the download button, we see that 3D structure is unavailable. Only 2D structure in SDF format is available. To convert the ligand structure in 2D SDF to 3D SDF format, we require the smiles format of the ligand structure. So at first we copy the smiles format of the ligand. Then we open the Avogadro software. Next, click the build option and select smiles format. Input the smiles format of the ligand which copied from the PubChem database and insert it. Next, click the export molecule option. Here you see that in the save option, the structure is saved in various format. Here we select the STF format and named as ligand.stf and save it. So here is the ligand. Next, click the OpenBabel app. After opening it, select the path where we want to save the ligand molecule in the PDB format. Next, select the ligand molecule which we saved in the desktop and convert it. So here is the ligand. Open the ligand in Bolivia Studio for visualization. So this is the ligand which is ready for molecular docking analysis.